Well, look, thank you so much. I mean, it's really exciting. I'm sitting here at 11:19 in the morning on a Thursday morning in scorching temperatures. Um, North of us, about 500 kilometers, is drowning. People are dying and being drowned because of excess rainfall. And so, like your continent or your country, ours is vast and variable. And in my humble opinion, we are seeing very much the effects of climate change, global warming, and more importantly, I feel that the fungi have a role for us to play in future life on this planet. So I am speaking from the land of the Bunurong and the Wurundjeri traditional owners. I pay my respect to the elders, past, present, and emerging, and extend that respect to all First Nation people who might be listening to this discussion, whatever country you're in. I acknowledge their continuing connection to the land and the waters, and I thank them for protecting this region and its ecosystems since time immemorial. Our First Nation people have been here over 60,000 years for continuous habitation. I finally acknowledge that the First Nations sovereignty of this land, Australia, has never been ceded, and Australia was, is, and always will be a First Nation land. I say that with great respect and great humility, and I hope that in my short time on this land, I can not do any harm and that my footprint will be light. If you have a look at this map, if you can see my cursor and you can see Melbourne right at the bottom, if you have a look at the distance from Melbourne up to the top of Queensland, it's about 3,500 kilometers, and I'm not sure what that is in miles. And then if you look from Brisbane on the east to Perth on the west, it's about another 3,500 kilometers. So we are a large country, and I'm talking to you from just down on the bottom right-hand edge of that circle that says Melbourne. I'm just about 20 kilometers outside the boundary of metropolitan Melbourne on a farm, which 60 acres, you're in acres, 23 hectares for us, 60 acres. Now, the Bunurong people, the Wurundjeri and the Bunurong people are people of the hills and people of the sea. And you can see the Bunurong in that pale green area at the bottom here. This is all their area, and we have the people, we have communities still here, very important to us, and we're learning more and more and becoming more respectful of our First Nation people. As I say, our First Nation people have been here for 60,000 years that we know of, and growing. And the originals, well, the history has been rewritten very badly in that it says that our Aboriginal people were nomadic, that they were not farmers, that they were not residents of particular areas, that they continued to move. And yet this isn't correct, because what I'm showing you here is evidence of their fishing ponds that they used and are still here today. You can see them, where they have been using and collecting fish and harvesting fish and eels as well. So they actually had a society that was settled in particular areas, even though they did move on because of the various weather patterns, and so they would move to different areas. But it is absolutely untrue to say that our First Nation people were not farmers, were not harvesters of produce in a farming situation. One of the grasses that they harvested is this wallaby grass, and there's great areas of it, and we're trying to bring it back as a native grass because it's found that the actual nutrient content is orders of magnitude greater than the introduced fodder grasses that we're told to use. When you look at the first settlers who came here, I call them settlers, and I use that word advisedly because I feel that Australia was invaded, but that's my personal opinion. When you look at the original diaries of these people, they spoke very much about finding great fields of the native grasses, and the seeds were being harvested for making flour. They found little caches that were upwards of a ton of this seed that was obviously put away for the season or out of season for making flour. So to say that our Aboriginal people were not farmers is absolutely incorrect. And we're hoping very much today that history will be rewritten and corrected. So I have spent my career looking at fungi. I have spent my career looking at what happens below the ground. I always feel, and I always say to my students, that if you look after and farm what's under the ground, then you can grow anything above the ground. And in most part, probably 10% of our mushroom kingdom or our fungal kingdom will produce mushrooms above the ground. And all the rest we tend not to see. We tend to see them as maybe white filaments on bark coming off a tree. We see them as potential spots and lesions on plants where they are causing disease. But only the sort of the big mushrooms do we see in spring and autumn here mostly, that are big and fancy. But all the rest are so important. And now 45 years I have spent looking at mushrooms, and I look down my microscope every day, and I am absolutely in awe because I will see something that I haven't seen before, or I haven't seen for 20 years, or I haven't seen for a long period of time. And the fungal kingdom is simply one of the major kingdoms, neither plants nor animals, but certainly in a kingdom defined of its own. And what's really interesting is that we're finding that the fungi were very, very early colonizers, because if you have a look at some of these fossils here in this one, it had to be fungi. It couldn't be anything else because there were no plants at this time. There were no other parts of the kingdom other than bacteria of the various kingdoms that could be there, and they're not bacterial. They're actually filaments, so they are fungal. So they had to be fungi. And if we look, we're going back 2.6 billion years that we are finding the evidence of fungal-like fossils. If you think about animals coming away, we're coming up 1.5 billion. If we're talking about plants coming from animals, again 1.5. But it looks as though that we've had our fungal associates here for well over 2.5 billion years. Now we have to respect them. And if you come right up to the top of this particular chart here, humans have been here for a blink. And yet we have caused such destruction in that blink. And I feel personally it's my responsibility to try and minimize that impact, because we don't know. We know probably 10% of our fungal species. We know, as someone mentioned earlier, about making beer from yeast. We make wine from yeast. We make cheeses from yeast. They've been there since humankind sort of had this serendipitous, left some nice grape juice in a bag, and it suddenly turned into wine, or someone left some milk in the sun and it's turned into cheese. These are serendipitous. This is the fungi telling us, as a human species, what they can do for us. There is a lot more now in technology and biotechnology where the fungi are absolutely essential, making tofu and some of the sake. I mean, they just go on. It's fungi after fungal species after fungal species, and so we must look after them. Also, I would suggest that if we stop taking coal and oil and gas out of the ground and started to look at a industrial size manufacture of fungal hyphae, that fungal hyphae can give us cloth. It can give us all sorts of things that we can use without having to mine the planet. So fungi have a huge importance, and I would put it to you that there would not be life on Earth if it wasn't for the fungi. So I have great respect, and I still love my fungi. I get up every day loving my fungi. It is now considered, when the first plants came from, well, when the first organisms basically came from the sea, the water, onto the land, the plants were ferns. Now, the ferns they don't actually have roots. So the fungi were here. There was no soil. There was no. It was just the bare rock and sand, silt, and clay. So there was no organic matter. So how are these plants actually living? And what we feel is that probably this is the first association of mycorrhizae, that mycorrhizal, that symbiotic relationship. That it was actually the ferns, the mycorrhizal fungi that became the roots of those first ferns and started harvesting nutrients from the soil, or what there was of soil. There was minimal soil because there was no organic matter. And they actually provided the phosphorus and the water and the carbon, etc., for the ferns. Then the ferns die, and then we start to build up organic matter. Then we start to build up soils. But that mycorrhizal association continued. We believe. We don't have evidence this far back, but in my literature search in time, I haven't found going back that far, but we do have actual evidence of mycorrhizal association with plants at around a fossil at around 600 million years, and so even that is awesome. Originally, all of the life on Earth was aquatic. And so when the first fungi came through onto land, they had these motile or these little tails, flagella. Now, I'm not actually showing you a fungus here, but I'm showing you a picture that will show the flagella that are on the sporangia. I'm using this term, Sam and Cornelia. I'm assuming everybody understands these terms if you're fungal people. I'm hoping, but I'm quite happy to answer questions if I get. I'm trying not to be using jargon.
Right. Well, we did, Dr. Cole, we did.
Call me Mary, please. I'm Mary.
Yes, Mary, we did say that people can type questions in the chat should they not understand something.
Okay. Well, thank you. Well,
so far, I think you're doing beautifully.
Right. Well, these little fellows, these little round things are called sporangia, and we still have them in the fungal kingdom, the first division within the fungal kingdom. And what happens when this particular they live in water? They have their little spores, which you can see floating around here, that come out of these sporangia, and then they will go and find a host. Now, as I say, this particular organism is no longer in the kingdom fungi, although it used to be. It's now in another kingdom with all the algae. But the actual mechanism and production of these little spores is the same. So the first fungi came out of the water, or the first fungi were in the water, and they had these little sporangia and their little sporangia spores and whatever floating around, but they came out onto land. And the mycorrhizal association continued as it has continued through today, except in areas where toxic chemicals such as superphosphate, lime, particularly manufactured lime, urea are put onto soils. One of the first things that happen when superphosphate is put onto soil, the mycorrhizal fungal association is damaged. And sometimes it's damaged irreparably. But Mother Nature is better than we are. She will repair if she's given the opportunity. So in all of my career, I have advocated no synthetic chemistry. We have our farm here, which I'll show you later on. We have been here for 47 years and we haven't used synthetic chemistry. And so what we're trying to build up, and the other thing I might say too, we've increased our organic matter in our soil from 12% to 23%. So this mycorrhizal association, it's like a network, it's a highway, it's a super highway. It connects trees. Trees are not individuals. One must never plant a tree by itself because it will be lonely, it will be sad. It must have the association in the soil to be able to function, because trees are communities. They're like villages below the ground, and we must keep that association going with the mycorrhizal fungi, moving from one root to another and connecting roots, and it's like a big telephone network. And it's harvesting the nitrogen and potassium and phosphorus from the soil. And in return, it says, well, okay, you know, I can't photosynthesize. I can't take my oxygen and my sugars from the sun. So let's have a contract here. The contract is, I will give you all of the NPK that you want and all of the sulfur and all of the water that you want, but in return, I want sugars. And so this relationship is mutualistic, one for one. I'll give you some food, you give me some food. We're all happy. We will all be healthy, and we can expand and have a beautiful lifestyle. So they're talking to one another all of the time. They're communicating. Trees are telling another tree when they've been attacked by a pathogen. They talk to one another. They put out chemical signals, and I fantasize a little bit because I just think, you know, all of this communication that's going on, how beautiful it is. Wouldn't it be wonderful if we could actually tune into that? Well, we can tune into it by being sensitive to what we do, wherever we are, in a forest or in our own backyard and our gardens. We can tune into it. We can listen to it, and we can be part of that community. This particular picture showing a little bit more of the mycorrhizal association. It is just so important. There are very few plant species that don't have a mycorrhizae of some sort, and they're mostly just the brassicas. But everything else has some form of mycorrhizae. If it's a proteaceae, then it's got its own proteaceous mycorrhizae. If it's an orchid up in a tree, then it has an association as well. Most of our trees, or all of our trees, have an association of some sort. And this photograph down here, you can see this amazing network. I mean, you put a plow through that, and you damage that network. This is what happens in cultivated land. There is no mycorrhizae because it keeps getting damaged. This, as I say, this started 2.5 billion years ago. It is so important to us having an environment where we can live on the planet into the future. And you can see over here this little one where mycorrhizal fungi, it sends up a little fruiting structure. Some of them are edible, some of them are really choice, but they disperse their spores, and so the life cycle goes on if we allow that to happen. And this is what I'm hoping you all do. Otherwise, I don't think you would be in your club. You all have like mind. One of the really important things that have been found quite recently, and that's by Sarah Wright when she was doing her PhD in the US. She was looking at a humus and accumulation of carbon and all of those sorts of things, and she kept finding in her experimental work a mucilaginous type nothing that she could not put a name on. It appeared to have no actual value, but as it turns out, it is actually one of the most important soil exudates coming from the mycorrhizal fungi in terms of how it makes life on Earth possible. And it was given the name of glomelin. And glomelin is an exudate that comes from just the metabolism of the fungal biomass, particularly the mycorrhizal fungal biomass in an undisturbed soil, more in an undisturbed soil than in a disturbed soil. And what has also been found is that it will actually hold more carbon than do the humates, depending on the soil type, but generally more carbon. Now, we have a lot of talk here in Australia at the moment, oh, we must be planting these particular types of plants because they have the capacity to hold a huge amount of carbon. But when I listened to these conversations, I said, but what about what's under the ground? You're not talking about what's under the ground. On the plant roots are the mycorrhizal fungi. The mycorrhizal fungi are exuding the glomelin. The glomelin is holding carbon, and it will hold carbon. It's in this whole carbon capture thing, it's not difficult to capture carbon. What's difficult is to actually hold it for a long period of time. And so if we've got a nice undisturbed soil, or a soil that has a 100% plant cover over it all of the time, where we have healthy, deep roots with a very active microbiology going on in the soil, then there's going to be truckloads of glomelin being produced, and therefore we're holding huge amounts of carbon. And so, to me, we shouldn't even be having to talk about this. It is so important for the future of the globe. And it doesn't matter what soil type, it doesn't matter what country we're in. This is so important. So it can capture carbon and it can hold it for us. But at the same time, that huge biomass that you can build up in the soil in a healthy sort of undisturbed system or minimally disturbed system, it's going to hold water. Now, when you think there's probably only 1% of the water on the planet that's potable, that's drinkable, that's usable, should we not be trying to find every way we can in our agricultural systems to protect and hold as much water as we can, so that when we grow our crops, and hopefully into what we call it pasture cropping, that we're holding all of that moisture in the mycorrhizal biomass, so that the plants are more resilient when we've got all of these changes in temperature. And it means that we can grow a good crop with minimal water, because we're holding a lot more water. So again, it comes back to the fungal biomass. It comes back to the fungi. They are absolutely essential to our agriculture. They're essential to how we can make the best use of water, which is, we don't make water. Water just glows around the globe. It's a finite resource. And there's so little of it that's available, that we have to be able to use it efficiently and effectively if we're going to keep a population which possibly will keep on growing on the planet. This shows you some of the mycorrhizal, the roots of a plant. If you've got a good soil where your roots can go down very deep, then they have the capacity then with their mycorrhizal fungi to be able to harvest a much larger area of soil, to be able to provide the phosphorus and sulfur and water and whatever it is to the plant whatever the plant happens to be. And so when you do put a spade or a shovel down into the soil and you can see your roots going down, well down into the soil, then you know that you've got a healthy soil as long as there hasn't been any synthetic chemistry put on it, because the roots are going down there, the soils have got a good texture, and the mycorrhizal fungi are really functioning and doing what they're meant to do. And if you dig up some of these plants, you can, this photograph that's down on the bottom right, if you just gently dig up a root and wash it, and you can use, if you're in a laboratory, you can use a stain. But if you're just at home, you can use a food coloring or India ink. You can actually stain up the mycorrhizal fungi. And then you take your washed roots, and you put them in some food dye, or you put them in some India ink, some black ink, leave them for a little while, then take them out and rinse it off, and then put it on a slide, and then put some water, put it in water, put your cover slip on the top, and you can actually see your mycorrhizal fungi. You can see your vesicles, and you can see these little arbuscules that are called here, they're like little cabbages. You can actually see them. You don't need great, high-powered microscopes or anything to see them, or lots of chemicals that are used in laboratories. You can actually do it with just ink. This particular photograph the diagram is just showing again the association with the plant roots. If you have a look at your plant roots there, if you've got good mycorrhizal fungi attached to those plant roots, which means there hasn't been any synthetic chemistry used in that environment, that root system of that tree can increase by 100% or more, which means that the actual amount of soil that the tree is deriving its nutrients increases by orders of magnitude. Then all of the nutrients are solubilized by the microbes, and then pops up your fungi that you go harvesting and foraging for in this particular season. So it all goes around if it's allowed to have a natural system. And it's so important, the fungi is so important in the decomposition of organic matter back into the soil that gives us our healthy soils and the healthy produce that we can ultimately have. And then just our healthy trees. If we don't have healthy trees and involved in respiration and whatever, then we're not going to have precipitation. Where there's no trees, you have the precipitation is down. Where you have a forest, you get lots of precipitation, lots of rain. So they are essential. But again, the fungi are involved. So this is what I'm saying. In the water cycle, if we want to have water and precipitation coming into our area, we've got to have forests. Take away the forests, we reduce the ability of the cloud formation and the precipitation to occur. So again, healthy fungi, healthy soil, healthy vegetation, a healthy planet, a water cycle that works for us. This one I use a lot because when I look at the bottom photograph, which is modern agriculture, I weep, because the destruction is total when you're looking at what happens below the soil. Up here, where we're using plows, there is damage, but you can see on the top right-hand photograph. Now let's go back. This one on the left-hand side, you see all the vegetation there, the grasses there. That is at the moment undamaged, and there's lots of mycorrhizal fungi there. And so they're doing everything that they need to do. The thing with the plow, it's going to do damage if we look at the top right. But if you have a look at each one of those hills, you can still see the green plants. Now those plants are still there, they're green, so the mycorrhizal fungi is still attached to them. Now it's been damaged, but not irreparably. Here, you've got green plants here, so there's mycorrhizal fungi, but when you have a look here, there's nothing. So that soil, that was soil, is now becoming sand, silt, and clay, because it's going to have synthetic chemicals put on it, They're going to grow a crop. They're going to take away the crop. They're going to put pre-emergence fungicides and pesticides. They're going to put post-emergence fungicides and pesticides. They're going to put all sorts of chemicals to grow the crop. But what that's done is basically all but destroyed the life in the soil, which does all of this for free. They don't send bills, you know, they do it for free. This is what they do in return for photosynthetics coming from plants. We can't go on and do this. This sort of farming is not sustainable.
Right.
This sort of farming is sustainable. And in Australia, we call it pasture cropping. So what we do is we say, okay, we have a pasture, a mixed species if possible, as mixed as possible, all across our paddocks. We then take some sort of a plow instrument and we simply open up the soil enough to plant our seeds, whatever those seeds happen to be. And then we allow that to close up, and in most cases we can grow a crop. There are still people who say you can't do that because a lot of the seeds have been modified and developed so that they can't cope with competition. Well, let's start to go back to something a little more natural that can cope with competition, because Mother Earth has diversity. You don't see monocultures in in natural systems. There's diversity. So it's not that we can't do it. Let's just think and change the paradigm and say the paradigm is that we are not the only species on this planet that has to survive. There are other species, and this photograph at the bottom is definitely not the way to go about it. I could talk for days on that because I get so angry.
So Mary, we do have a number of questions that are building up in the chat.
Yes.
And so I was wondering if we could.
Yes.
Okay. By the way, with the pasture cropping, is that called a disc harrow?
But one of them was there. Yes. Yes.
All right. So someone was asking whether there is equipment currently available to do this kind of farming on a large scale.
Yeah. Yeah. Okay, I'll answer that one first. If you Google, there's a gentleman, a lovely gentleman in Brisbane, Queensland. He has developed a plow called a Yeoman's plow, Y-O-E-M-A-N-S, because his name's Yeoman's. What that is a series of plate, like discs, that go in and cut into the soil, but they don't turn over the soil. They simply lift and aerate the soil. Now, there's another one. There are several things like that, but the Yeoman's plow will give you an idea of what I use. We use a Yeoman's plow on our farm here, and it just discs in. It just cuts into the soil at an angle, and usually we do a pass of say 100 millimeters, and then we do a pass of 300 millimeters. And then we plant, we don't grow crop, but we do grow multi-species pasture. And it goes in, but there are people who are growing wheat, barley, rye, corn, maize, that have done it this way and got crops without any problems. So what the instrument actually does is it has a plate, and it has the tines that come down and go through and simply dig into the soil, but it doesn't turn the soil over like a plow. So there's several of them, but look up the Yeoman's plow, and that will give you an idea of what those of us who use those sorts of things, and the croppers who use those sorts of instruments, that's what we're talking about. So it's a case of not turning over the soil, simply opening up and aerating, but allowing the pasture or whatever the plant is that you've got growing across the whole surface, to allow that not to be damaged so much, so that the mycorrhizal fungal component will come back and close up that gap where the seeds are.
All right. It looks like someone posted a link to something similar to Broad Forks. I'm not sure it's a link to the Yeoman's plow, but someone else had a question about how can we, and I think you're going to be answering this, but how can we encourage the desirable fungi to grow in our gardens?
Yes, I can, because I have real problems with the word desirable, because
Okay.
all fungi have a role. So I will address that. But what it comes back to diversity. We will never know in our lifetimes how many species of microbes there are, how many bacteria, how many fungal species there are. And so in nature, we have diversity. And so what we must do with our soils is use composts and compost teas and minimal disturbance to try and keep that diversity there. If we keep the diversity there, then every fungal and bacterial species is beneficial. Because I have a talk that's coming up at the with the Soil Food Web, where I say pathogens are of our making. Every organism has a role, and I'll talk about that a little bit later. So I don't use the word beneficial or bad or good. They're all good.
Okay, great.
They're all good.
And then another person had a question about, and I love this, I love your do-it-yourself, use food dye or India ink. They had a question about the process to using these inks and low power microscopes to see what happens.
Yes, to see if you've got a good mycorrhizal association, like an arbuscular mycorrhizal association, the roots are clear, and often you can boil them in vinegar or something like that, just to, if you've got any tannins in them, and then put them in the India ink or in the food dyes. You only need a microscope that has 100 times mag. So let's assume you have 10 times eyepiece and you have 10 times objective. Then you can actually see the vesicles in the cleared plant tissue. That you don't need high power microscopes. If you can go up, 40 is actually too high. I prefer a times 20 for all my fungal work. And so here in Australia, I tell my clients who are interested in using microscopes to get rid of the 100 times oil, because you don't need it, it's a nuisance, and replace the 100 times oil with a 20 times, and then have a times 4, a times 10, a times 20, and a times 40. And you'll find, if you're interested in fungal community, you use the times 20 more than anything else.
Huh, that's so interesting.
And so I've actually got a supplier of microscopes here in Australia. I mean, they are imported, but I like them. And so I think I'm probably his best customer, because my clients come and say, please, what microscope do I buy? So I send over to Steve and I say, I need some microscopes. So I get a discount as well, which I pass on. But what I also have asked him to do over the time is please get rid of them 100 times and replace it with a 20. And he does that at no extra cost. So 100 times oil is just a nuisance, because unless you can use it correctly, you end up with oil all over your other objectives, and then you have a totally stuffed up microscope, and you can't see anything. Doesn't matter how good your prep is. So get rid of the oil. Replace it with a times 20. You'll find that you'll use the times 20 more than anything else. When you're looking at your mycorrhizal fungi, you should never have to go above a times 10.
That is lovely to know, and
yes,
much less complicated. Thank you so much for that.
My pleasure. My pleasure.
I think we can move on now.
Okay. Well, the next photograph here is an example of how we grow lettuce. Here, not far, within 10 minutes drive of me. I'm in a big agricultural area here, and this is how lettuce are grown. And the only positive thing about this method of cultivation is that one of the agronomists was my PhD student, and he's since finished and passed successfully. He's incorporating a lot of compost into this particular farm and trying to, if not maintain a good mycorrhizal association there, he's at least getting the organic matter back in so that the microbiology, if it is there, has got something to live on. And he's removed in very large parts of this particular farm any synthetic chemistry. So relying on just compost tea, fish kelp and molasses, and good compost. And so kind of in a commercial situation, he's doing his best. Let me put it that way. He's doing his best. He doesn't own the farm. He's simply an employee there, but he's doing his best. And I have to give him 100% for trying. We then look at some of the organisms. The fungi have actually changed the course of history, very much so. Now, this particular organism, the Phytophthora, is no longer in the kingdom fungi, but the Irish potato famine probably put a lot of Irish people here in Australia, certainly a lot of Irish people, I think, went to America at the time. And the famines were just all consuming in that country. But why? Have a look at the photograph. It's monoculture. You have one plant, you have one organism, you have absolutely monumental destruction because there is no diversity there of plants and different organisms to be able to mitigate this particular Phytophthora. Here where we are, we grow a lot of asparagus. And we have found that we have Phytophthora cinnamomi here, which is causing a lot of problems in our forests, particularly. But I had a local farmer come to me and he said, "Look, I am just losing so much of my asparagus crop to Phytophthora. What can I do? What chemicals can I put on?" So I said, well, if you're wanting to put on chemicals, you need to go find yourself another consultant because I don't use chemicals. However, if you would like to try compost, then I'm very happy to help you. So what he did was he finished the season and we loaded compost into that soil. Now, we didn't get rid of the Phytophthora, but what we did do was he was able in the next season to get an amazing crop of asparagus. And so we've continued to use the compost and we're gradually getting rid of the Phytophthora through competition, diversity, microbes, other fungi. It can happen. You can do it. I have spent a lot of my academic career looking at the wine industry, mainly because they funded all my research. So I was very, and plus I enjoy a nice glass of Shiraz, I might say. But what's really interesting about the wine industry is that we have all of these diseases, so-called diseases, caused by mostly fungi. And I might add to you guys too, most of them came from America. So thank you very much. And they have around the world, they've gone around the world, botrytis and powdery and downy mildews all came from America. Thank you very much. And so created a whole lot of problems. But again, we have a monoculture situation. And so a lot of these organisms, they're there. But if we had the grapevines growing as they do, as they used to, and not in monoculture, then we can work with these organisms. We can work with them. And I have vineyards now who haven't used synthetic chemistry and fungicides and things in 25 years because of the way they manage their vineyard, the way they plant their cover crops and things like that bring in lots of beneficial microorganisms and or not so much beneficial, but just diversity of organisms as to be able to compete with some of the pathogens. And the pathogens are only pathogens because we've allowed them to be because of monoculture. They have a right to be there. And so it means as the more diversity one can have, then the more competition there is from all of the microbial world, which means that those organisms that think they're going to cause some damage or try to cause some damage, they're being kept in check much better by all of that diversity. And Botrytis cinerea is an organism that I spent much of my career working with. And it's an amazing organism. It's a fungus. It likes high sugars, so it comes in at around the time that we're harvesting our sugars, harvesting our grapes, and therefore it rots the fruit. Well, if you have a think about the original grapevines, they come from this area that's in yellow, Georgia, between the Caspian and Black Seas. They grow up into trees. They're a vine. They grow up into trees. The fruit is way up in the canopy of the vine. How is the grapevine going to reproduce if its fruit and seeds are up in the canopy of the trees? It has botrytis to rot the fruit, drop the berries, release the seeds, new generation. So if you think about botrytis from that point of view, it's absolutely essential. It's not a pathogen. We've made it a pathogen. It's absolutely essential for the recycle or the cycling of the grapevine life cycle. So, you know, don't tell me that pathogens are a problem and we have to get rid of them. We have to rethink the way we have our agriculture. So are pathogens of our making? Yes, absolutely. And botrytis is a perfect example. It's there, it has a job. And just because we want fruit that's absolutely pristine to make wine, then we think it's a pathogen because it rots our fruit. Well, we have a little saying here in Australia, stiff cheese. No, we have to change the way that we use it. This is how vineyards, grapevines originally, not necessarily in this forest, but in this sort of forest, and you've got diversity there. Then you take it across to here as to where we grow our grapes today. Monoculture, we wonder why we have pathogen problems. Lots of chemicals put, well, I'm putting it to you that my grape growers don't use chemicals, that they manage their vineyards very, very carefully. So again, it's management, it's working with nature. We don't have to throw synthetic chemistry out because it's easy. Here's a vineyard. This young man was one of my students, and here is a vineyard that I call a wonderfully fertile, diverse vineyard. Their wines win trophies all around the world. And if you look out, if you can find Crittenden's wine, then I would suggest you buy it because it's some of the best. But look at the vineyard. It has lots of diversity. It has grasses. The chefs plant broad beans up the mid-row so that they can harvest the broad beans before they get worked in and mulched down. This is what I call a healthy vineyard. This is what I call a healthy environment. This particular vineyard is full of beneficial insects and spiders and everything that helps to keep the vines healthy and clean. It's full of a huge diversity of fungal species. And so the actual pathogens, the ones that we call pathogens, don't have an opportunity to establish in more than just a minute amount, but not enough to do any damage. And so it's absolutely doable. Cornelia and Sam asked me to show you our property. Here is a drone photograph of our property, but unfortunately it's summer and it's very dry. We still have a good grass cover, but we have 60 acres. And if you can see at the top of the picture here, we have these plantations all the way along our boundaries and in between the paddocks, which means we have shade for the cattle and protection from the wind. Now here in the center, this is our home. We have this here is my laboratory, this particular building here. And it was initially built as Alan's shed, and I had a little room in it. And then I had another little room. And then I had another little room. And I said, I need this whole shed. So I got the whole shed. So it was gutted, and I now have a laboratory that takes up this whole shed. If you notice this shed at the back here, Alan has recently built this shed. If you also notice, there is a little fence around it where he keeps his farm equipment. Inside that fence, I'm told, is a Mary-free zone. I'm not allowed in there because if I see it, I'll take it over. So he's not giving me the opportunity. Down here at the back, you can see we have very little hay. Because we don't need it. We have our stock numbers at a stocking rate where we never ever damage our vegetation. We have, even in this dry rain, dry conditions, we still have plenty of grass. It's just dry at the moment, so we rotate our stock very regularly. We have water that comes from bores in the ground, and then they have lots of protection where they can keep out of the sun and the heat. So we don't. This little bit of hay really is just a backup. Down here in among the trees, we have another area, another shed, which is called Hen Hilton, and it's where our chickens live. And so our chickens also, everything on this farm is basically organic, and we have a very large veggie garden. You can't see terribly well here, but just along here between the end of basically my lab and Alan's shed, there's an area here that's it's all covered with wire to keep out the birds, and it keeps out the rabbits. We have rabbits. You know, that's a thank you very much from the British. So we have a lot of pests that have all been imported. So we have a huge veggie garden, and so we basically during COVID, we really didn't have to go to the shops because we had all our own fruits and veggies. Up here, if you look on this side of the house, where you can see the 2 water tanks, just in this area here, we have an orchard of about 15 different citruses, and so we keep ourselves in juice every morning with fresh juice. The vegetable gardens, just at the moment, we have tomatoes like you wouldn't believe. Everybody who comes past our door, we sort of hijack them and say, please, would you like some tomatoes? They're organic. You know, we can't get rid of them. We have passata, we have soup, we have everything. So that the chickens now are getting big meals of tomatoes because we can't keep up with them. So this is sort of our particular farm. We have glass houses both at the garden near Alan's shed and here near my lab, where we grow out of season. And so, yeah, this is my little patch of paradise. I love it here. We've been here 47 years. We've not put a chemical on it, and we're very happy here. This is from the other side. This is another view. You can see Hen Hilton down the back here, where the chickens are. They have in amongst the trees. They're able to. They've got. We have foxes here as well. Thank you very much from the British. So we have to keep our hens locked locked up at night. But they have little areas of fenced area in amongst the trees where they can go and graze and get their worms and things. And then you can see the garden here with the cover over it a lot better. We have it covered with wire, but we have wire that birds cannot get caught in. So it's a very specific diameter for the holes so that the birds can't get caught. And again, just looking terribly dry at the moment. This is our north boundary. So again, we have vegetation. And then also in this area here, we have our beehives. So we have 2 or 3 beehives. So we have the lovely. So all of the pollination goes on in the garden here, and then we have this lovely beehives as well. Just another view. Now this is coming down to ground level. As I said, and this is our boundary. Do you see this yellow line here? This is our boundary. This is the next door neighbor where it's yellow. We have not used chemicals, synthetic chemistry. What we use is compost tea, compost, and we use fish hydrolysate, kelp hydrolysate, and we use low sulfur molasses in our compost tea. And so if you can see our grass, it's nice and green. Down at the bottom here, you can see our lovely Angus animals grazing happily. But if you see here, we have a weed called cape weed. It's a broadleaf weed that grows a yellow flower. Stock will eat it for a very small time of the year, but they don't normally like to eat it at all. You'll notice that our neighbors don't grow grass, they grow weed, cape weed. And here you can see from another view, cape weed. And then you see our place, no cape weed. Why is that so? When I do a soil test on the area where they're growing the cape weed, it's bacterial dominated soil. When you have a look at our soil, it is fungal dominated soil. And so once you get a high amount of fungi, particularly mycorrhizal fungi, the weeds are outcompeted. The grasses and the medicinal plants and the various diverse species that we have growing in our pastures, they outcompete these weedy species. But the major thing that you notice is the difference between the fungal domination of our soils and the bacterial domination of the other soils. Now, these people have lived next door to us about the same time as we have, but if you have a look at this bottom photograph here on the bottom right, do you see when Alan's is really naughty? When he puts the compost tea out, he just has a little spray about a meter into the next door neighbor, just wondering whether they'll notice that particular meter inside their boundary doesn't have any cape weed in it. They've never bothered to ask us why. But you can see here, it's absolutely evident just outside the boundary. Do you notice there's no cape weed? Again, if I take a soil sample just in that little area just outside the fence, then you'll find there's fungal domination. And I can tell you this, Elaine Ingham can tell you this, but if you see a photograph like this, it is so blatantly obvious that by using compost and compost tea and no synthetic fertilizers, you can get a result like this. And it doesn't cost you anything. And it's not doing any harm to the soil. It is so easy. Here is another one looking north. This plantation here is where we have our beehives. But this is, and there's the plantation, that's the edge of our property. You can see the lines where the Yeoman's plow has been through. Alan will go through, just open up, aerate, and then he'll come through with his compost. Well, it's all done in one pass. He has the tractor, he has the Yeoman's plow. On the back of the Yeoman's plow, he has his compost tea container. Each of the tines that go into the soil, he has a little dripper going down, and the compost tea, as the tine opens up the soil, then the compost tea goes right down into the bottom, whether it's 100 millimeters or 300 millimeters. I'm trying to think about what that is in inches, say 4 or 5 or 6 inches down to 20 inches or whatever. The compost tea goes down in, so it means that he's able to feed the compost tea with the fish kelp and molasses down into the soil, and then as it closes over, you get all these lovely responses in the grass. But that's all we do. We don't do anything else. We don't put anything else out. Just compost tea with the additives in it, and we've done that for 47 years. And as I said, we've increased our organic matter from around 10 or 12% when we came here. And the last time I did it was a year or two ago, and it was 22, 23% organic matter. Here's some more views of our this is Alan's. This is the Mary Free Zone. Do you see the fence? That can't get through that. These are more of our plantations, and down here is Hen Hilton. So again, even when you look down at ground level in the summer, we've still got some green grass. Now, this is one of our front paddocks. This is again one of just the fence that's around one of the paddocks, and there's Alan's shed there as a reference. About 25 years ago, we were given some rock phosphate that came out of the ground from China, and we put down a series of 1.5 meter by 30 meter long plots just to test this material. We put down equivalent of 100 kilometers per hectare, 200 and 400. Now, 25 years later, do you see this green? That is that same material that was put down 25 years ago at 400 kilograms per hectare of rock phosphate, totally unprocessed, just mined, and it's still there. We're still getting a response. And the microbiology activity in that area is amazing still. Here's a vineyard. And again, I'm showing you by example rather than, but compost tea. You can see at the end of the vineyard, you can see up in the headlands here, you can see all of the capeweed. You can see the pale yellow of the grass. But have a look here where he's been putting on compost tea, which we've been making for him. Do you see how much greener it is? Do you see there's no capeweed? Do you see how much greener it is? There's no capeweed. And yet out here where there's no compost tea, there's capeweed. Now, when you do a soil sample, this is fungal dominated, this is bacterial dominated. So the compost tea, my growers we make the compost tea, or they make the compost tea. They spray their vines to run off so that it goes into the ground. This is all mulch in here. They've only just pruned, so the mulch is just about used up here. So what they'll do is now they'll add more compost, more mulch into this area coming through the winter, and just leave it until the spring. But you can see the difference. The vibrant green versus the pale yellow of the bacterial dominated soils. It is again very easy. Now, making compost tea. This is Alan. This is my husband. Everyone says, "Oh, compost tea is it takes time. It's difficult." Well, we went to our local hardware shop and we got a 20-liter bucket. And then we got some of the orange stuff it's like a plastic stiff pipe that is used in the electrical industry for putting electrical lines down. Heat it up, make it into a circle the size of the base of the bucket, put holes in it, go to a pet shop where they sell fish and get a little fish bubbler for aerating fish tanks, put it on, you've got a compost tea bag, and there's your 20 litres of compost tea. Now, all of that cost us about $25. And you can make compost tea. Now, if you make a really good compost tea, you can dilute that. You can dilute that down to 250 litres of compost tea if you want to. But it's not difficult to make compost tea, and it's very easy, and it's a lot of fun to make it. So it's easy to be made. Our big commercial 1,000-liter compost tea brewer is this big stainless steel tank, and we make compost tea. And you can see the two compost bags here with the compost in them, and then there's fish and kelp and molasses and whatever in here bubbling away. And so people will come to us and say, look, we just don't have the facilities to make enough compost tea because we need large volume. Would you make some for us? And I said, yeah, of course we will. That's, happy to, but you have to come and get it yourself. But then all of a sudden, Alan says, haven't been out on the tractor for a while. Well, here's a compost tea brew coming up. He has to have an excuse to go out and have a little ride on the tractor. So he makes his brew of compost tea and puts the tank on the back of the tractor, and off he goes, as happy as a bird, putting out his compost tea. So he's got 2 or 3 hours on the tractor. So he has his little play on the tractor, but he's put out some nice compost tea, comes back, washes everything out, and is very happy that he has had not only a little ride on the tractor, but he's actually done some good for his pasture. Does anyone want to ask questions now? Because I'm going on to another.
We do have a few questions. And so someone was asking about the makeup of your soil, which I think you partially definitely answered with the fungal dominance versus the bacterial dominance. And they were also asking about the kind of texture of your soil.
Well, we have a really good friable texture. We would have what would be called a peaty loam. So it has a small amount of clay in it, but it's mostly a loam because we've increased the organic matter so much. We have a loam. When you actually walk out on the pastures, it's actually quite spongy. You get this lovely give. We don't have compaction because we're very, very careful about how many cattle we run. And we're very careful about if we happen to have some very dry weather or some very wet weather. If we have very wet weather, then we'll try to look at where the wettest part of the property is, and we'll close that off until it drains, so that we don't get the pugging, the footprints of the animals. If it's very dry here, we don't have the compaction because we have protected this soil for 40 years. We have resilience in the soil that our neighbors don't have. We don't have any dust. We don't lose the grass cover. We don't have any dust because first of all, we don't allow the cattle to eat the grass down to that extent. But also because we've protected the soil for so long and we have such a huge fungal biomass in the soil, we hold water for months after, longer than people around us. So we have never had dust because we've always had 100% grass cover. Mm-hmm. So to answer the question is, our soil, you'd probably call it a peaty loam because we're continuing to increase the organic matter all the time.
So people also have asked about worm castings, or whether that's possible to incorporate that into what you do.
Absolutely. Yeah, I like vermiculture as much as I do thermal composting. A lot of people have their little worm farms in their gardens and put it out. Then the people who have broader acres, some of them make commercial amounts of vermicast. Very good. The only thing that I do find sometimes that it can be a little bit bacterial. And so often what we'll do with the vermicast, the actual solid part of it, not the liquid, but the solid part of it, we'll use it and incorporate it into a lot of woody material and make a compost windrow out of it. But absolutely, I mean, worms are good. Worms are good. If your soil doesn't have worms, then it's not good soil. So worms are good, and anything to use their great capacity for recycling, then you do it. So yeah, worms are good.
And then I'm curious, did you base your compost recipe on anything in particular? One of the things here for a lot of people that are trying to be more conscious of our environment and compost is they always say, don't put dairy, don't put oil, don't put meat, don't put whatever. I have found that with bokashi composting, doing the fermentation composting, that you can put meat and oil and dairy and it composts just fine. But so I'm just curious about your particular recipe. We also have somebody asking about we have a lot of leaf litter, we have a lot of pine needles, we have kind of that sort of material that we could work with too. So
yeah, anything that has ever lived can be composted. You just have to look at changing your recipe. Now in here, you have pines. In Australia, we have eucalyptus, and we have another group called the Melaleucas, and we extract oils from them because they have medicinal qualities. They have antiseptic qualities. So what we do with the pine trees, which are introduced species in Australia, and our eucalyptus and our Melaleucas, if they come down and we're able to mulch them, then what we do is we lay them out in long windrows, probably certainly no more than about 1.5 meters high, and as long as you like. And we turn them with the bin that's on the front of the tractor. We turn them over regularly until we can no longer smell the volatiles. And once the volatiles are gone, then it means that most of the antimicrobial oils that are in the pine or the eucalyptus or the whatever, then it means they've volatilized off or they've been leached out from the rain. Only then would we put those materials into our compost. Because the whole thing about those oils is they are antimicrobial, but we're wanting to grow microbiology, but it doesn't mean you can't use them. It just means you have to put them aside for a couple of months, or depending on the season, maybe 6 months, and then they're admirably usable. In terms of using protein, protein is a nitrogen source. But when you put protein into a compost, you've got to be aware that protein may also have organisms like Salmonella or E. coli or something like that in them, which adds a level of sort of occupation, health, and safety issue to your compost. So what we do if we're going to use protein, I do a lot of work with the pig industry and the chicken industry, and much as I loathe this factory farming, they have to deal with their mortalities, and it's heartbreaking to see the number of mortalities in this factory farming business. So what we do with those is you can make your windrows, you can put your mortality, and I suppose if you come back to a smaller level when we're just talking about gardens, but let's look at this big level because it tells you that it can be done. We ask them to set the base of their compost pile up with lots of straw and brown woody material, mulch. Then to lay their mortalities side by side, not stacked, just side by side. So this would be the same thing with your kitchen scraps in the middle of the windrow, so that there's a good couple of feet. I'm having trouble with the translation. A couple of feet on either side to the outside of the windrow, and then on top you then start to build your pile again, so that the protein, whatever the protein happens to be, is well encased in the center of the windrow, and then you put your green material and whatever, so that you increase the heat in the windrow and start the pasteurization process going, and then we find if we don't turn then for a couple of weeks, particularly with the chickens. In a couple of weeks, all we've got left is skeleton. About 6 weeks after that, we have no evidence that there were ever chickens there.
Wow.
But they have to come up to temperature. They've got to come up to 65 degrees at least 4 or 5 times to be able to do that. They get turned at that time. Now, when we're looking at the big mortalities like pigs and cattle, we tend to have what we call static piles. So we'll set up the pile in the same way as I told you, with maybe 2 feet of straw or something like that. Then we put our mortalities in the center of the pile and then cover it. And then on the outside, we have about 2 feet, there's 30 centimeters, about 2 feet, I think that would be, of a cover on the outside. And then if you're going to make a static pile, you can actually cover that with soil and then not touch it for 12 months. And if you just leave it there, when you come back, you will have the most beautiful compost. You will never know that there was a mortality in there.
This is
so
really
everything can be composted. Now, if you come back to the kitchen situation, then in your small pots, there's lots of different ways of composting. You have your little bins, and there's all sorts of digesters and things. But if I just like to work with nature, and I work with just little windrows or little rings of compost. What we do, if we're going to put protein in, we put it right into the center of the stack where it's hottest. And then we use our green material for our bacterial food, our nitrogen, and we use our brown material for our fungal component. We put the protein in the middle because that's nitrogen as well, and that's where the stack is going to get hottest, quickest. And so your decomposition of that protein material is most rapid, and usually at the first turn, it's not there, it's gone. It's gone. Now, in terms of fallen leaves, like you would have loads and loads of leaves. What we do is either rake it up and then we go through with the lawnmower or a mulcher and break it all up, and then that becomes part of our brown fungal food, a part of our compost. And we use sort of 45 green, 55 brown, thereabouts, to make our compost. I had a workshop last weekend, and the compost pile went from 18 degrees, which was ambient temperature when we made it, at lunchtime on Saturday. It was 64 degrees at 4 o'clock on Sunday. It has now gone up to 65 degrees 3 times, and it's only, for us, it's Thursday.
And are you talking Celsius or Fahrenheit?
Oh, sorry, Celsius. Sorry.
Well,
yes.
Okay, so that makes a big difference.
Yeah. Celsius. I don't know what the conversion is for you. I'm sorry.
Maybe somebody can put it in the chat.
Yeah, but what it means is that you don't want to get your compost heap any higher than 65 degrees. That's the maximum temperature. So whatever that converts to Fahrenheit. But what we've done, we made it Saturday, so Sunday, Monday, Tuesday, Wednesday, Thursday, five days, and our compost pile that we made is now down to probably 70 to 80% of the volume it was 5 days ago. And that's a very active compost pile.
Wow.
So, it's very doable. So it's getting that relationship between the nitrogen and the carbon at about of the bacterial food to the fungal food, which is nitrogen to carbon. We look at for the compost that we make for Compost Tea, we look at about 45% green material, nitrogen material, and about 55% brown material, carbon material. And so Alan picks up all the twigs that fall from the trees, he puts them through the mulcher, it's all used. Yeah, how am I going for time? I must be running out of time, am I?
Well, we often run till 9 o'clock. We do have people who run a little later. And I don't
want to go over time.
I know you have a lot of information, and we had a few more questions just about how you apply the compost tea. I think that was one of the devices you showed that Alan was using, how often. And then also someone had a question about, why did you not apply the rock phosphate everywhere? And I imagine you ran out of it.
Well, we did ultimately. Mm-hmm. We did ultimately, but we put it out across the whole property at about 150 kilograms per hectare, because we got a nice response. But that 400 kilogram, that's 25 years ago. It's still there. It's still there. Now, composting, what we do because we make highly fungal dominated compost tea, you have to be very careful putting it out, because if you've got bends or you've got filters or anything, it gets caught up. So what we have, and it's actually on our website, and our website is agpath, agpath.com.au. And over on the right-hand side of the homepage, we have an information section, and in there we have these. What all they are is just a bar, a copper pipe about what's that? Is that 6 inches? About 6 inches long, and about a couple of inches in diameter. And then at one end it has like a little bar, and it comes straight out of the tank, and then depending.
Uh-oh. Let's see. Mary?
She's frozen.
Okay, so the signal must have dropped.
We've
been doing great coming all the way from Australia.
At her side, yes.
Yeah, so
so she
now
may drop
the
out.
Presentation.
I have it, but it's not her talking about it.
Okay. Well, I'm hoping that she can figure out to come back in. I know we have
time for everybody to take a bathroom break.
True. Oh.
Okay, so she's dropped out, so she's going to join back in.
Right. All right. Thank you, everybody that's here. This is really fascinating.
And thank you for your questions.
Yeah. I wanted to ask, so if that Melaleuca pile, the eucalyptus and all the Melaleuca tea tree stuff is antimicrobial, can you bury the pigs and chickens in that and have it duke it out, before you end up with losing all the volatile oils? And then the other thing is that I think actually my discussion on logs to food with Joe of Field and Forest and Patrick, they're talking about how you can take pine, which is not normally useful for growing things, but degrading it enough to a certain point where it can be used to make really good potting soil and replace peat. So that was another discussion too.
That again?
That composting pine chips and conifer chips, that actually doing it over a long enough time can be a replacement for peat. Right now peat's not a sustainable source of potting mix, etc.
Right. Yeah. Cool. Thank you.
So
is there a good place to get compost if you don't make your own?
does anybody want to answer that question? I mean, I do know that like DeKalb has like a free actually, where you can pick stuff up from them, but you don't actually know everything that's gone into it. So I don't know.
But that's also to make your own compost. She may be asking about just buying compost ready to go into the soil.
Does anybody have any answers to that?
I have bought mushroom compost from the garden shops, and those work out real well. This mushroom
There's
compost.
Mary, just let her in.
Yep.
Yes.
Yep.
I have something too, Cornelia. There's some companies that pick up
Oh.
compost in the city.
But there's not, okay, she's getting,
Hello.
Yep, I need to make her a co-host again.
We just had a power outage.
that
goodness.
makes,
I'm
okay.
sorry.
No. We had a little discussion. So Karen and some of the other people, if you can post some of that information in the chat for people, that would be great.
Can you
And
still, can you still
Mary,
see my,
I've just made you co-host again.
so can you see my talk?
It's not up yet. You would have to bring it back up.
Oh, heavens.
Have faith.
Yeah, okay, I don't know how to do that.
Can you put the copy that you have in, dear?
Uh,
We had, so Mary, Sam may be able to insert the copy that you sent him.
yeah,
So we
so
could-
just hold on, share screen. Share screen. Here I'm going.
Oh, there we're seeing,
Are you coming? Is it coming up?
Yes.
Yes.
Right
Okay,
now we're
okay.
seeing old wives' tales.
Okay, that's it. Okay, just let me slideshow. No, I don't want to go from the beginning. I want to go.
Click on the slide.
Oh, there you go. Amanita
that
muscaria.
was the slide you
Beautiful.
were on.
From current slide. Okay,
Excellent.
there I am. Sorry
Great.
about that, everybody. We had a power outage.
Okay, no problem. We're, whenever you're ready, Mary, we can
Oh,
start.
okay. So the next part is the mushrooms, which is probably the part that you're all most interested in. This Amanita muscaria, it's imported into Australia. It's not native in Australia. It grows only on the introduced species of pine and larch, silver birch, some of the imported species. Mycorrhizae, obviously, as you know, and just quite beautiful. It comes up for us in our autumn, and because we have many pine trees, often as trees in parks and along streets, we have these amanitas growing along the roadways. And of course, people are always terrified of them because they feel that, you know, little children or dogs will pick them up, which is always a potential problem. But again, it's an introduced species for us. But what we found is over the last, if you think about the introduced pines, they were introduced into we're going to go again, I think. I think we have an unstable system. We've got a storm coming up, would you believe? Electrical storm, but no rain. So if you lose me again, I do apologize. 150 years ago, because we don't have softwoods here in this country as such, or usable softwoods, pines were introduced from all countries around the world to see which ones we would be able to use as commercial timber. And so at the same time that they came in, so did a lot of the mycorrhizal fungi come with them. So we have our pine, we have our these types of mushrooms only associated with those introduced species. But around about, we're not exactly sure when, but if we say round about 50 years ago, in Tasmania, our island state, it was found that the Amanita muscaria was growing actually on the native vegetation there, on the eucalypts. So in probably 100 years, which is no time in mycological time, they had naturalized and crossed hosts. And so we're seeing a lot more of that now, of particularly the Amanita species, because they're the ones that people worry about in terms of being dangerous to animals and people and whatever. We're finding that they're crossing hosts in different places. Up in Canberra, which is our capital city, it's in the middle of New South Wales. We have Amanita phalloides in the oaks there that are starting to cross hosts. No, it's not working for me. I'm not able to change my screen. I'm sorry, I can't change my screen. It's not changing on me.
I think you could talk about whatever you want to talk about next, even if it doesn't change.
Okay, then. Hold on. Rachel, I don't know what's happening. So the Amanita phalloides, which is another mushroom that has come in with the oaks that were imported into Australia. We're finding there, again, oaks that are around the 40 to 60 years old. As you know, mycorrhizal fungi are successional. So the ones that you start off with in a young tree are not those that will be in mature trees as such. We were finding that the Amanita phalloides was starting to show up at around the 30 to 50 years. But at the same time, it was starting to cross hosts into the eucalypts. Now, about 1990, I was invited to be a mycologist with the Poisons Bureau. And since then, I still do a lot of work in mushroom identification. And what we were looking at when people would phone in and say, I think we have, we've got a poisoning, we could always say with the Amanita phalloides, well, is there an oak tree somewhere nearby? Is there some sort of introduced species somewhere nearby? And then we could say, well, okay, if it looks like a mushroom, just an agaricus, but it's got white spores and it grows in association with one of the European trees, we could very carefully and succinctly say, well, it probably could be an Amanita phalloides, therefore, treat accordingly. But what's happening now, when it's crossing over into the eucalypts and into our own vegetation, it makes it very difficult for those of us who are consultant mycologists to be able to pull out Amanita phalloides, for example, which is the most toxic of the mushrooms and the one that's most easily confused with an edible mushroom. We can't do that as clearly anymore. It's added a real problem for us. And we haven't actually had anybody die, or maybe just the odd person, from eating Amanita phalloides, but they've picked it up accidentally, because they're out foraging for the agaricus, the field mushroom, which is a nice, and I wish I could get my slides to move. I don't understand why they're locked up.
I can, if
Could you?
I can go ahead. If you stop sharing your screen, I'll share my screen. You should be able to see it.
Okay, where do I find the stop sharing? Okay, stop share. I'm stopping sharing.
Okay, now.
I don't understand why it's locked up. So if you can go, I'm so sorry about this, everybody.
So Mary, do you see that now?
Yes, I do. I do.
Okay.
Now,
so this was, I was at Amanita muscaria, or
yes.
and then
we
this
go
is
to
what
the
your
next
next
one.
slide was.
Yes, and it's really interesting. The magic mushroom, psilocybe. We have a lot of psilocybe here in Australia, and it's used as an alternative psychedelic by a lot of people. It's also there's a particular restaurant, or there used to be a restaurant up in New South Wales, where you could go and ask for your steak and mushrooms. And if you ask for your steak and mushrooms in a very particular way, then you got a really nice little meal of the psilocybin as your mushroom component of your sauce. And so they've been a part of history forever, and they're now being found. It was interesting, the conversation that they're now being used and looked at very carefully as possible medicinal. And I'm sure they have, because the ancient people, the previous civilizations have used them for years. So we're just starting to discover again. So I'm sure they have got medicinal positive responses. Okay, next one. The one that we have most problems with is this Amanita phalloides, because although this particular picture shows it being very yellow, they often can be quite pale and very similar to Agaricus campestris. And if you just went and picked that and didn't have a look at the bulb that's in the vulva, the little sac that's in the ground, and just pulled it off or cut it off, it would look very much like a field mushroom, except that the gills are white. And so what's happened with the poisonings in Australia is the people have gone out and foraged, and all but one or two of their mushrooms have been the Agaricus campestris, the field mushroom. But they haven't looked carefully at their underside, and they've picked up an Amanita phalloides. And of course, the Amanita phalloides, the effect of Amanita phalloides is disastrous. And there was one that I was particularly involved in. A lady ate, she had eaten it on the weekend. She showed sort of food poisoning effects within 24 hours. She went to the doctor. She was told to go home and take a Panadol. A couple of days later, she ended up collapsing. She was taken into the hospital. I was called and asked what I thought, because the family was asked to get a mushroom that she had eaten, an example. It was put in a taxi cab and it was taken to me at the university. And as soon as I saw it, I said, it's Amanita phalloides. Well, at that stage, she'd gone into hepatic failure, but they were able to treat her. And she was in hospital for many, many months because they were able to protect at least some of her liver. And of course, as you know, the liver is one of our organs which will regrow. And so the lady ultimately left hospital. But the doctor said to me, and I'm sure he was exaggerating, and he said, look, she only had a tablespoon of liver left, but we were able to save it. So the fear now is that the Amanita phalloides has moved into the native forest, and it's not as easy now for people to be able to pick them. They have to be much more careful. Next one. Lots of old wives' tales. I'm sure you have the same thing. If the poisonous mushroom, it's poisonous if it turns a spoon or a coin black. Well, no, that's not right. If it's eaten by animals, then it's safe. No, that's not right. If it tastes and smells good, well, no, that's not right either, because Amanita phalloides tastes amazing, apparently. And a lot of our little insects and slugs and whatever eat our mushrooms, but they're not edible. The other one that really makes me laugh is if it peels easily, it's safe. Well, most mushrooms do peel easily. Hello, a bit of nonsense there. One of the other ones that we end up with, most of the people who end up in hospital in Australia during the mushroom season, is this two Agaricus species here. Do you have a yellow staining mushroom over there, Agaricus xanthoderma or similar? What we have, do you see the one at the top here? You can see that when you bruise the cap and the stem, there's a little bit of yellow shows up. We have a group that we feel are probably 4 or 5 species, which we call the Xanthoderma Group. And they have an alkaloid in them that's very much like the phenolic materials, or the materials that are used to wash hospitals, and smell absolutely disgusting. I don't know how anyone can cook them and still eat them, but they will make very, very sick, but not life-threatening. So we kind of just say to the doctors when they phone up, we just say, look, they'll sort of feel as though they'll wish they'll die, but they won't. They will survive, but this is the most common poisoning that we see here in Australia, are the yellow staining Agaricus species. Next one.
We do have them here.
Yeah,
in North America.
Yeah. So here you can see the yellow stainer here. This is what we try to have people look at when you go out and forage. Just gently score the edge of the cap or the edge of the stem with your fingernail. Put it in your basket, lay them out in your basket, and then before you go home, go and check all of those where you've just done your little bruise, and any of them that are yellow, put them back. They have a role. They've got every right to survive. Put them back. And then just take the ones that haven't got a color, okay? Now this one, do you have the Chlorophylla molybdenus in over there?
We
This
do.
is one of our more common poisonings.
Yeah, we had
Yes.
poisoning last year, actually.
Yes. Well, the lepiotas, the macrolepiotas, and some of those have changed generic names as well. But they're very common. The two are very common. Not so more up in northern New South Wales and Queensland, we see a lot more of the chlorophyllum. And I get a lot of pictures coming down to me asking for identifications because we find it here, the lepiotas, we pick them, and because they're kind of big and robust, we tend to cook them and put them away for the winter for winter soups. But it's really important that when you've got something where you've got potentially poisonous species, it's very important to drop spore prints. I always tell people, if they're looking to forage, start to get a habit of dropping spore prints so that you know what color the spores are. And in that way, you can often do a lot more identification yourself rather than having to go to, you know, some expert or to the herbarium and whatever. But that one is, yeah, that one's a particularly bad one for us. Okay, next one. These ones again are come into Australia. They're not naturally found here. They are imported. And the same way with the boletes, the Swillius and the Swillius granulatus and luteus, they've been brought in as well into the pine plantations. But what we do find, you know, I tell people, when you go out and forage, take what you want for yourself. Don't clear fell the fungal flora. Just take what you need and leave the rest. But we've been out into some forests where people have gone out to pick to sell, and it's just like a clear fell forest. It's tragic. So just take what you need. These are beautiful to eat and pickle. Next one. Well, again, events that have caused, I talked about Phytophthora, claviceps, St. Anthony's fire. During the Middle Ages, contaminated rye was a major problem with certainly with armies. So again, fungi involved. Another one. This was interesting. Chernobyl. Mushrooms were used to be able to monitor the fallout of radioactivity from Chernobyl, and right up through Scandinavia and across into Europe. People were out monitoring the mushrooms for the level of radioactivity, and this is how they were able to monitor the cloud of radioactive fallout. So again, mushrooms, you know, helped us out in this terrible situation. Next one. So I guess mushrooms, not mushrooms, fungi are the reason that we're able to live on the planet. Most of the fungi are in the soil, and those that come above the soil spend most of their life in the soil. And so what I like to think is, I have spent my career sort of under the soil. If we look after the soil, if we look after what's in the soil, if we look after the whole environment in terms of not poisoning the soil, then we have the capacity to leave something for our future generations. Next one.
I believe that. Oh,
that should be,
yes.
That should be the end. Everything that I've shown there has been acknowledged here from somewhere or other or somebody or other, and I thank them for their use. Thank you, everybody. I'm so sorry about that bleep.